Hello everyone. So yeah, it's very exciting article, and I would like to share you some results from here. So we start uh, from agenda for today. We talk a little bit about B cell changes here. Then we move to two main, I think, parts of this article. It's T cell changes and inflammaging theory from researchers. Then we move to epigenetic analysis and validation of findings on human samples. So first of all, experiment design. Researchers have two group of mice. First, it's from 3 to 4 months young group, and second, it's from 70 to 24 months. It's an age group, and there are three ways they analyze data. They isolated few organs here, then perform bulk TCR sequencing analysis from them. Second way, they sorted CD45 plus cells. Basically, it's just immune cells, and perform on them single cell methods like single cell RNA-seq, single cell TCR-seq, BCR-seq, and ATAC-seq. And third way, they validated findings using flow cytometry data here. So let's start with the B cell changes. First of all, they show us that percentage of B cells from all CD45 plus cells remain stable in two groups here. But if we look at the single cell RNA-seq data, there are this interesting area here and here. It's age-associated B cells that were described previously in several articles. They have this phenotype. And it's interesting because if you can see here, the number of these cells dramatically increase during aging, and it's also validated on flow cytometry here. So two more interesting things that within these age-associated B cell subsets, there are two subsets. So it's a peritoneum B cells here and spleen B cells here, and they have a quite different expression profile. So, for example, peritoneum B cells express high level of this ZCVPV1. It's a zinc finger protein that enables methylated histone binding activity. And spleen age-associated B cells here and here, they mostly expressed FCRL5. It's a member of the immune globulin receptor superfamily and the FC receptor-like family. So both of these subsets expressing another zinc finger protein here. It enables DNA binding, transcription, and repressor activity. So next, a few words about B cell receptor analysis and T cell receptor analysis. So just to remind, so they calculate few metrics, very interesting and useful metrics for repertory analysis. It's a clonality and B cell receptor mutation frequency. So why it's so important? So as you know, B cells can recognize antigen, and during this antigen recognition process, they activate and proliferate. So during this activation and proliferation, they can mutate the B cell receptor. It leads to more efficient recognizing of B cells of antigen from B cells. So during this process, we can measure clonality and its increase due to proliferation of B cells. And B cell receptor mutation frequency is also increasing due to this mutation of B cell receptor. So here is the repertory analysis, and they reclusterize B cells of RNA-seq data, and two main clusters here. It's a cluster number 3 and cluster number 7. It's a peritoneal age-associated B cells and splenic age-associated B cells. So if you look at the clonality level of each cluster, we can see that two main clusters that are increased in clonality. It's a cluster number 3 and cluster number 7 in aged group. But if you look at BCR mutation frequency, there are only increase in cluster number 7. but not in cluster number 3. It's very interesting, and it seems to be an antigen recognition process in peritoneum and splenic in spleen. So it's interesting why it's so and what antigen they can recognize here. So here, quick summary for B cell sections. So we saw that there are increase of splenic and peritoneal B cell subsets, which are H-associated B cells. And also there are increase in clonality of H-associated B cells. And the splenic H-associated B cells have increased mutation frequency. So next we move to T cell analysis and start with CD4 plus T cells. Here, they analyze two main subsets. It's FOXP3 plus CD4 T cells, which are regulatory T cells, and PD1 plus FOXP3 minus CD4 T cells, which are mainly exhausted CD4 T cells. And we can see here that percentage of FOXP3 CD4 T cells increased only in spleen of age group mice. But if we look at exhausted CD4 T cells, there are increase of this subset of cells in all organs. And also, if we look at the clonality analysis, so we can see that clonality remains stable. So this change is not sufficient. You can see it on the y-axis. This change is just normal. And one more interesting metric for like repertoire analysis, it's a shared number of shared clones. Basically, it's just same clones that we can find in a few organs. So here, uh, everything is normal. So all unique T cell clones are within a unique organ. So there are no antigen recognition or something like that. Next, we move to CD8 T cell analysis. Here, clustering of CD8 T cells based on localization. And here is some subsets of CD8 T cells that researchers are identified here. And we can focus on this area here. You can see that the number of age-associated T cells here, it's a PD1 plus CD8 T cells, dramatically increase during aging. And two more points that they try to characterize expression profile of these age-associated T cells. And they have very high expression of granzyme K here and tox factor here. And of course, also PD1 plus. So next, few words about granzymes and PD1. So granzymes, it's very important proteins in like cytotoxicity of CD8 T cells. So due to recognition of CD8 cytotoxic CD8 T cell of target like cancer cell or infected cell, they can produce a lot of perforin and granzymes that are leads to apoptosis of target cell. So basically, it's a main like enzymes to kill target cells. And PD1, PD1, it's most important thing in cancer therapy, I think. So why it's so? Connection of PD1 and PDL1 in on target cell, it leads to like exhaustion of CD8 T cell, inhibit proliferation of CD8 T cells, cytokine production, and the cytotoxicity. So permanent activation of this pathway, it leads to very high exhaustion of T cells. Let's move to another analysis here. They try to like compare age-associated T cells with some data sets and subsets of known CD8 T cells. For example, here they took a public data set of CD8 T cells from mice that are infected by LCMV virus. It's a very like aggressive virus for immune system, especially for T cell immune system. So it leads to very high exhaustion of all T cells. And here you can see the different stages of exhaustion of T cells. And last stage is terminally exhausted T cells. And if we look at the profile of age-associated T cells here and terminally exhausted T cells from public dataset, we can see that there are some similar expression profile. For example, expression of granzyme K here and here, of PD-1 and tox factor. So they very similar, and it seems to be like very close subset of T cells. Next, we move to clonality analysis here. And if we look at the clonality of CD8 T cells, clonality increased in all 4 organs. And also, if we look at the number of shared clones, there are a lot of clones that are present in few organs and identical T cells that are consistent and preserved in all 4 organs. So if we look at the clustering of different clusters by phenotype on single cell RNA data, so there are few very interesting clusters. It's cluster number 4 here, here I guess, cluster number 4, 8, and 9, and they express very high granzyme K and yeah, granzyme K here. So if they calculate clonality based on clustering, so we can see that clonality increase in cluster number 4, 8, and 9. And we can say that this increase of clonality is just due to increase of clonality and expansion of age-associated T cells, but not other T cells. So yeah, here is a quick summary for CD8 for T cell analysis. So proportion of PD-1 plus FOXP3 minus CD4 T cells increased almost in all tissues. Also, expression of PD-1 on CD8 T cells marked the emergence of distinct cell subset that was virtually not presented in the NIH. And they characterize phenotype of associated CD8 T cells, and they was granzyme K plus tox plus PD-1 plus cells. And also age-associated CD8 T cells phenotypically very similar to terminally exhausted CD8 T cells. So next, inflammaging theory. First of all, they perform ligand receptor analysis that that can reveal some interactions between ligands and receptors on different cell subsets. So here they see that on granzyme K plus CD8 T cells, there are very high expression of A4B1 integrin, which is CD49D. And they validated these findings on flow cytometry here. You can see that activation of CD49D increased in age-associated T cells. It's a red bar plot here, and even in blood. And then they see that, and they saw that A4B1 integrin connected with fibronectin-1 especially, and other markers here. But most interesting part is fibronectin-1, because fibronectin-1 is expressed in macrophages, dendritic cells, and fibroblasts. And next step, they try to show some connection with pro-inflammatory phenotype of fibroblasts and granzyme K expressing CD8 T cells. So here it's public data set of fibroblasts, and they show us that fibronectin-1 expressed in fibroblasts here, 3 clusters here. And most interesting part that expression of fibronectin-1, so fibronectin-1 is co-expressed with the main pro-inflammatory cytokines of fibroblasts. It's interleukin 6, CCL2, and CXCL1. And also, to validate these findings, they performed 2 more experiments here. So they took 2 groups of fibroblasts. One, it's a normal fibroblast here, and senescent fibroblast. So they exogenously added some stimulation, for example, granzyme K or granzyme K with interferon gamma, and calculate activity of interleukin 6 or CCL5. So they show that the additional stimul of granzyme K is not enough for normal fibroblasts to boost their pro-inflammatory potential. So, but if they perform it on senescent fibroblasts, they show that granzyme K is just enough to boost very important cytokines, interleukin 6, CCL2, and CXCL1. So here it's a inflammaging theory from researchers. So there are some CD8 T cells that during aging differentiate or accumulate in different tissues, and they have phenotype TOX plus, PD1 plus CD8 T cells with the high expression of CD49D. And via connection with fibronectin 1 on macrophages, dendritic cells, or different other cells, they come to tissue and express very high amount of granzyme K, which stimulate normal cells or senescent cells to produce a lot of pro-inflammatory cytokines, such as interleukin 6, CCL5, CCL2, CXCL1. And here, the key role of inflammaging they give to like CD8 age-associated T cells, which express granzyme K. Next, they try to identify how these age-associated T cells like accumulate or differentiate, or what happened with CD8 T cells in age environment. So they took two group of mice. It's a young donor and two hosts, one young host and aged host. So donor cells have CD45.1 plus immune cells, and host cells have CD45.2 plus cells. And it's very important to divide where is the donor cells and where is the host cells after transplantation of immune cells. So they transfected CD8 T cells from young donor to young host and aged host, and number of transfected CD8 T cells was the same. But if you look at the if we look at the number of age-associated T cells after transplantation, the number of age-associated T cells increased only in host age host. So here you can see like about like 20%. But if they perform this experiment vice versa, so they transfected CD8 T cells, and mostly they were age-associated T cells here, 36%. It's a lot, I think. So they transfected them to young host, and it's about 1% transfected CD8 T cells. But there are no differences in age-associated T cells here. So the number of age-associated T cells not increased, or nothing happens here. So taking together these results, these results show that development and expansion of CD8 T cells, it depend only on cell extrinsic age-related factors, and that differentiation of CD8 T cells remains stable in like young environment or old environment. So it seems to be very like stable subset of T cells. And here there is some summary. Age-associated CD8 T cells interact with fibronectin and other ligands here via connection with A4B1 integrin, which is CD49D. Also, granzyme K induce secretion of pro-inflammatory cytokines in fibroblasts. And a theory has been put forward about the inflammaging, where the key role have H-associated CD8 T cells via expression of granzyme K. Development and expansion of H-associated CD8 T cells depend on cell extrinsic age-related factors. Let's move to epigenetic analysis. So they clusterize ATAC-seq data and defined 15 distinct clusters here. And the main interesting clusters, it's cluster number 9, 11, and 12 here. They were profoundly expanded in aged mice compared to young group. And these clusters, it's the same three immune cell clusters that changed with age. It's H-associated B cells, cluster number 9, CD8 H-associated T cells, cluster number 11, and T regulatory and activated CD4 T cells, cluster number 12. And they identified some motifs and transcriptional factors that are enriched in each cluster. So for example, for CD8 T cells, there are increase of T-box motifs here. It's a T-bed family of transcriptional factors. For T regulatory and activated CD4 T cells, it's a BATF family of transcriptional factors. And if we look at the CD8 T cells, there are two main transcriptional factors for CD8 T cells. It's T-bed and EOMs. And it's interesting that expression of EOMs was elevated, whereas the T-bed was decreased in CD8 T cells. And these results suggest that different role of T-bed and EOMs on CD8, mostly in H-associated CD8 T cells here. So it's interesting about their function in H-associated CD8 T cells. So here is a quick results about epigenetic analysis, that there are three main clusters: H-associated B cells cluster, cluster number 9, CD8 T cell cluster number 11, and T regulatory and activated CD4 T cells cluster number 12. And CD8 T cells demonstrated a strong enrichment with T-box motifs, and T regulatory and activated CD4 T cells demonstrated a strong enrichment with BATF motifs, and H-associated B cells with a strong enrichment with PO2FA motif. Then they try to validate the findings on human data, on human blood samples. They took 14 young people, 14 young individuals and 14 old individuals, and perform quite same experiments. And here the results, some general results about CD8 T cells number and granzyme K CD8 T cells. So if you look at here, it's like basic result in all articles before, where CD8 T cells uh, were were increased only in young group of individuals. But if you look at granzyme K plus CD8 T cells, there are increase in old group individuals. So then they, for granzyme B, it's just like normal control. There are no increase in granzyme B expressing CD8 T cells. And next, they saw some interesting connection that granzyme K plus CD8 T cells positively correlated with plasma level of key pro-inflammatory cytokines, interleukin 6, TNF alpha, and interleukin 8. Next, they try to characterize phenotype of H-associated CD8 T cells in humans. So here they clusterize data based on site-off analysis. And here cluster number 3, which is similar to H-associated T cells with expression of granzyme K and some other markers. And they saw here that there are increase of expression of CD28 and 27 and lack of CD57 here. And the number of this cluster, percentage of this cluster was increased in old individuals. So they called this granzyme K plus T effector memory cells that are expressing CD27 and 28 and lack of CD57. So next they tried to characterize granzyme B expressing T cells because it's just the normal CD8 T cells with normal level of granzymes. And they saw that here with clustering CD8 T cells, blue it's granzyme B expressing CD8 T cells, there are quite different subsets within this big subset. Here we can see that there are some cells that expressing CD45RA, and there are some cells that expressing CD45RO. And it basically means that there are T effector memory cells and TEMRA cells within these granzyme B expressing cells. So if they exclude from analysis naive CD8 T cells, they reclusterize data here and get that these granzyme B expressing CD8 T cells divided into two groups. Group that expressing KLRB1, it's a receptor of natural killer cells, and IKZF2, it's a TEMRA subset of cells, and this gene, it's transcription factor that involved in regulation of lymphocyte development. So then they perform pseudotime analysis to see differentiation of this free subset of cells from T central memory cells to granzyme K cells and to granzyme B cells. So they were initially associated with increase of granzyme K expression here, then followed by a shift forward expression of granzyme B. So in normal humans, there are granzyme K expressing CD8 T cells. It's just like second stage of differentiation of CD8 T cells. So the final stage is the granzyme B expressing CD8 T cells here. So it's very interesting why in old individuals this differentiation break down and or altered somehow. So maybe this increase of granzyme K expressing CD8 T cell is just accumulation of some second stage of CD8 T cells. And also they profile analyze epigenetic profiles of each CD8 T cell groups and define some key transcriptional factors for each of them. So for myeloid cells, there are enrichment of this motif. For naive cells, there are enrichment of TSF7. For central memory, there are enrichment of RANX1. And for granzyme K and granzyme B cells, there was enrichment of T-box and BZIP motifs. So yes, this is it, what they get from epigenetic analysis. Nothing interesting actually, just some facts from here. And here the summary for validation on human samples, that granzyme K plus CD8 plus T cells positively correlated with plasma levels of pro-inflammatory cytokines, and granzyme K plus T effector memory cells expressing CD27 and CD28 and were lack of CD57. And they identified two distinct cell populations within granzyme B plus CD8 T cells. It's TEMRA-like cells and T effector memory cells that are expressing KLRB1. And they identified a very interesting transition from T central memory to granzyme K expressing effector memory cells to granzyme B expressing effector memory cells. And this differentiation was associated with increase of granzyme K expression, and then with granzyme B expression, which is very interesting why it's so. And yeah, key results from my whole presentation that they confirmed increase of age-associated B cells during aging. They also identified age-associated CD8 T cells that were expressing granzyme K, PD-1, and TOX plus genes. Then they show us that producing of granzyme K by CD8 T cells stimulate secretory senescent-associated secretory phenotype to produce pro-inflammatory cytokines. And also they show that CD8 T cells, age-associated T cells, likely to be key players in inflammaging. And in humans, they have phenotype of CD28 plus, CD27 plus, CD57 minus subset of cells. And it might be breakdown in differentiation of CD8 T cells to granzyme B expressing CD8 T cells that leads to accumulation of this age-associated phenotype of CD8 T cells. Thank you, everyone, and join ComputAge Journal Club chat and Telegram to continue discussion. Thank you. Yeah,
Yes.
question.
Thank you very much. Thank you very much for this talk. Yes, Erik. Okay, this is a end of the talk, and we are starting discussion session during half an hour. Yes, as I remember, Vitaly.
Yes, we already have a question in chat.
Yes. If Sergey allows, I will read the question. If granzyme K is elevated, does it mean that all subjects analyzed had cancer cells or had pre-cancer phenotype? Question to the speaker.
No, it means just that they have some antigen that are persist in all organs. For example, it might be cytomegalovirus or Epstein-Barr virus that are very common in all tissues and all cells. So it's I think it's not cancer cells or a pre-cancer phenotype.
Okay. Who else want to ask questions? While others are thinking, I want to ask small technical question regarding slide 25. Could you show it, please? Yes, I wondered, do I correctly understand that CD45.1, CD45.2 is a kind of different splicing for variants, or this how should I interpret this to
it's
here?
a allelic variant of this like marker. So on on donor cells, this allelic variant is CD45.1. For host cells, it's CD45.2.
These are not splicing, these are genetic variants of the same receptor.
Yeah.
Thank you, I got it. And I have a question about pseudo time analysis. Could you show this slide? Is this with this brilliant picture? Yes, that one. If I correctly understood, according to this pseudo time diagram, we have a sharp transition from expression of granzyme K to the granzyme B subpopulation. I wonder why this transition is so sharp. So we have all cells of granzyme K plus in the left part of this parabola, and then we have only granzyme B cells. Maybe I misunderstand something.
Yeah, it's interesting, but I don't know why it's so. Just I believe to researchers that there are some very like sharp transition between these two subset of cells. Yeah. Uh,
But is this sharp transition corresponds different batches within the same single cell data set?
I didn't find information about that. Yeah, I think it's not technical batch, I think. Like,
Okay, but this double transition from TCM to TMK to TMB is this transition corresponds to how it could how to say that another way of asking it, if this pseudo time corresponds to aging process, increasing pseudo time corresponds to increasing age.
They think so. So they said that they think that this transition is just correspond to aging because in as I remember correctly that this transition granzyme B is a normal granzyme that expressing all CD8 T cells in normal like antigen recognition process. But for granzyme K, there are no interesting information, some differences between function of granzyme K and granzyme B. And actually, granzyme K is more, there are some information about granzyme K that this granzyme is connected with like tissue damage, a lot of tissue damage, not killing target cells, but a lot of huge tissue damage. So they assume that this connection is very clear. So granzyme K is like main main marker of aging in old individuals, and this transition is like breakdown in old individuals, and that's why so active process of inflammaging in these individuals. So I think they saying that it's a key problem here.
But you said that granzyme K is a hallmark, not a hallmark, but a biomarker of aging. The presence of granzyme K could serve as a biomarker of aging, right?
No, okay, from this article, it seems to be like that. Yeah.
Yes, it's interesting because according to the first blue diagram here, granzyme K cells are corresponds to low values of pseudo time.
Yeah.
And this, if I correctly understand again, this low values of pseudo time corresponds to low ages.
No,
No.
I think
no.
Pseudo time is just a trajectory in the middle, based on the average expression of cells. So it's kind of a weight to add time to some developmental trajectory, and pseudo time doesn't know in which direction it goes. So usually it's combined with RNA velocity, but RNA velocity also gives wrong directions. So the direction should be chosen by the researcher. And they actually have an option in this pseudo time, like reverse the direction or not. So they've chosen this.
But Alexander, do I correctly understand? But that pseudo time here is opposite to direction of aging, or all of these cells are of the same age?
I think here is the cell. So from like
sorry, could you repeat?
I think we didn't know. So here we don't know information about age here in this plot.
Okay.
Here is just mix of different cells and no connection with age just here. It's a more logical like connection researchers give here.
Okay. Now I see. Yes, then that's really another thing. Okay. Maybe somebody have question because I have one more, but I
Yes, I
will give you
have
a
one
chance.
question. So, Erik, thank you for your wonderful presentation. I have a question. Did they discuss how these results could be used in aging, to treat aging, or in further development?
They don't discuss about treat aging, but they discuss about like more about CD8 age-associated CD8 T cells, and so about functions of these cells. So why they so like high in old individuals, why they expressing granzyme K, and so on, so on, so on. They didn't talk about like treatment more. They more wondered about function of these CD8 T cells. Yeah. So I hope I answered your question. Thank you.
Yes, thank you. To understand it right, that external factors act on T cells, and external factors determine the old phenotype. Or is it like internal or external?
Yeah, based on these experiments, they show that it's external factors of age, like environment, can affect on all cells, and yeah, more likely it's about age environment, not cell specific, like some factors.
Yeah, that's interesting. So maybe we should focus on T cell receptors. So probably they have some receptors overactivated, like, or
yeah, it's discussable
okay.
because there are so it might be about so during aging there are a number of like naive T cells that could recognize a lot of antigens decrease during aging, but the number of these like exhausted percentage of these exhausted T cells that are tired and cannot like provide their function, the percentage of these cells increase due to decrease of normal and naive T cells. So maybe it's just like due to decreasing of normal cells. So so yeah, we didn't know about like T cell receptors, and I think it's not about like T cell receptors or something like that. I hope so.
Thank you. And one more question. I don't know much about T cell biology, but as I understand, naive T cells, they are progenitors of terminal or effector T cells. And terminal T cells are actually killer cells that are killing old cells, for example.
Yeah, but few like additional information about T cells. So naive T cells, it's a like CD8 T cell that didn't find yet antigen to like recognize antigen, and after that they like become effector memory T cells or central memory, and so on, so on. And after like some actually after their function, they might become exhausted T cells, so they produce some cytokines or kill some cells, and after that they might be exhausted, and T cells just do not perform some functions. They just circulate and cannot kill cells, cannot do some important job for the immune system. So actually they undergo apoptosis, but in old individuals they accumulate somehow. Yeah, so
Oh yeah,
that's
I see. Thank you. And how are they eliminated? Do you know? How are terminal exhausted T cells eliminated from the body?
Actually, I think that another CD8 T cells could kill them, but I didn't know. Yeah, I think it's another CD8 T cells.
So does it mean that the problem is naive T cells? So
It's
a lack of
Yeah, but we can't do much about that because it's due to like thymus um, atrophy. So during aging, thymus like become more fibrotic. So because of that, the output of naive T cells from thymus is decreased. So if we like just I don't know, transplant the new thymus, maybe it can help to immune system, but I didn't know. So how yeah, how solve this problem?
Okay, thank you.
Thank you for the question.
One more question. What's the average lifespan of naive T cells relative to organism? I mean, in mouse and human.
Average life, naive T cells. You mean
lifespan? I mean, is it comparable with my lifespan, or is it much shorter?
So
how
I think
many days, how many months do they live?
I think naive T cells live about 9 years, I think, if I remember correctly. So quite long.
Okay, and so is this an average lifespan, or I mean, what's the longest life?
It's up to 9 years. So
Up to 9.
maximum.
So this is the maximum.
Yeah.
Okay, thank you.
Thank you.
I also propose to discuss in more details their inflammaging theory. The slide with theory is formulated quite well. Yes, this one. This is quite interesting proposition that, if I correctly understood, we acquire some TOX plus PD-1 plus CD49 T cells, or some kind of progenitors of CD8 plus T cells, which then emanate granzyme Ks by inducing senescence, by inducing the expression of different cytokines and so on. This is quite interesting. And my question, it's a question, it's an open question for the discussion. TOX plus PD-1 plus cells, which are depicted here, where does they come from? How CD8 plus T cells generates them? What could be the mechanism?
So I think the mechanism here is, as I think I mentioned, about exhausted T cells. So because of exhaustion is the normal state of CD8 T cells after they perform their function. So in normal cells, in young individuals, this kind of cells just eliminated from the organism. by the other cells or some other ways. But in old individuals, they just accumulate because I think organism don't have such resources to eliminate all like damaged or exhausted cells, and they maybe accumulate in this state in all organism and perform this like damaged phenotype. Yeah, I think it's just accumulation of like normal cells, but they must be eliminated from the organism.
Do authors suppose that accumulation of this particular exhausted T cells is the only or maybe sufficient factor for inflammaging, or it is one of the many factors?
I think it's one of the many factors, but in this article they focused only on these CD8 associated T cells. Yeah.
Yes, they embed this factor within the modern inflammation theory.
Yeah.
This is
yeah, it's not so simple. Just one subset of cells could like produce inflammaging. I think there are some other.
this could be too simple to be true.
What do you think? Could it be the other way around? I mean, if the aged person doesn't have enough granzyme K activity, and this is why all certain cells cannot be removed. And instead of inhibiting granzyme K, I need to increase the activity of these proteases and related proteases.
I think according to this, the experiment here, so they show that in normal fibroblasts, in normal cells, there are not enough just to produce granzyme K to like boost pro-inflammatory cytokines by other cells. So here they said that
and by normal, what did they do? Did they have a biopsy, or was it a tissue culture fibroblast?
Yeah, it's a tissue culture.
Well, this is kind of completely
yeah.
different. I mean, in case of biopsy, that would sound clear.
Yeah.
But with tissue culture, it's very, very different and disturbed,
I agree
traumatized.
here. Yeah, I agree here, but if we talk just theoretically about granzyme K, I think it's not enough to like produce granzyme K, and so as I said, that granzyme K is produced by senescent cells. So senescent cells accumulate in normal individuals, but they like eliminated from organism very fast, I think. So this amount of granzyme K that they produce, it's not enough for like for senescent or for this, I think, inflammaging like process. So yeah, I think it's not enough to granzyme K leads to inflammaging.
All right.
Thank you.